This is Unit 14, where we talk about the miscellaneous gastrointestinal pathogens and methods to identify those types of pathogens. Today we're going to talk about things like Vibrio cholera, Vibrio parahemolyticus, Yersinia intercolitica, Aeromonas, and all these fun organisms. So stay tuned for more in Unit 14. Here we go. Yersinia intercolitica causes an infection similar to Salmonella. We don't see it in the United States too much, but we do see it more commonly in Europe. It is known to contaminate blood products also and can tolerate refrigeration. This organism prefers cooler climates like northern Canada and the northern United States. This organism can be a gastrointestinal pathogen, although it's not routinely screened for. It is a member of the enteric family. It's this species that may cause gastroenteritis, not Yersinia pestis, which is what causes the Black Plague. Okay, humans are exposed via contaminated food or water, so if they come in contact with fecally contaminated food or water, they could actually get infected with Yersinia intercolitica. It is distributed widely in nature, both in aquatic and animal reservoirs. And the organisms are very sturdy and can tolerate refrigeration without the loss of organism recovery. That's why it is an organism that is known to contaminate blood products. It can actually survive refrigeration. Okay? The optimal growth occurs at 25 degrees Celsius, and the media that we're going to set is CIN. The selective media is for Yersinia intercolitica is known as CIN, although it will also grow on MAC as well. Whenever we're trying to isolate Yersinia intercolitica, we always want to set up a CIN auger. It is selective for Yersinia intercolitica, and the organism appears as a bullseye where it has a pink center and a clear border. That is very typical of Yersinia intercolitica. And again, we do want to attempt to grow these at room temperature. They grow at 35 degrees, but they prefer 25 degrees Celsius. When identifying Yersinia intercolitica, we can look at its metabolic reactions to identify it down to the species levels. It is a sucrose-positive organism, so the TSI is going to be acid over acid. The KIA, which only has two sugars, it actually has lactose and glucose, is going to be alkaline over acid because it's the sucrose that makes the TSI positive. They are H2S negative and urea positive. Lysine decarboxylase and adenine decarboxylase are negative. Ornithine decarboxylase is positive. It is motile at 25 degrees, but not motile at 35 degrees. And we can identify this organism by simply setting up an API or some other rapid system like Microscan or Vitek. It's very easy to confirm this organism with API. Some of our pathogenic species can produce different types of toxins, and those toxins are either going to be neurotoxins, cytotoxins, or enterotoxins. A neurotoxin is a toxin that causes muscle paralysis. For example, Clostridium botulinum produces the most potent neurotoxin known to man. It's the same toxin that's used for Botox. A cytotoxin is a toxin that destroys intestinal epithelial cells. One such example of an organism that produces a cytotoxin is Clostridioides difficile. And then we have enterotoxins, and entero basically means from the intestine and then toxin. So an enterotoxin is a toxin that comes from the gut. Usually this is produced by organisms such as Salmonella or Shigella, and those enterotoxins alter the metabolic activity of the intestinal epithelial cells. Vibrio cholera is the next organism I want to talk about, and especially because we are in the midst of a current pandemic. This is a pandemic that's been going on since 1961. Several 100,000 deaths have occurred because of the pandemic in Africa, India, and actually 2.9 million people are affected every year by Vibrio cholera. Okay, this is a toxin-producing bacteria. It's been the cause of 7 pandemics, with us currently being in the 8th pandemic. It causes the most severe diarrheal illness. So this would usually cause a person to lose gallons of fluids within 24 hours. It is characterized by vomiting and rice water stools, and what they'll see is flecks of mucus and tissue in the stool, and it looks like grains of rice. A patient can actually go into shock within 12 hours of the initial symptoms. We don't usually culture for Vibrio cholera, but we always look for beta hemolytic, cytochrome oxidase positive organisms on blood agar because those could be Vibrio cholera. Shock and death from this organism can occur in 24 hours because of the fluid loss and cardiovascular collapse. These patients that have Vibrio cholera lose gallons of diarrhea every day. A person gets Vibrio cholera from contact with contaminated food and water, and we do need an enrichment broth that is alkaline and buffered to a pH of 9.0 in order to isolate Vibrio cholera. Then we can take that isolated colony and put it on thiosulfate citrate bile salts agar. In thiosulfate citrate bile salts agar, this differentiates our sucrose positive strains of Vibrio from our sucrose negative strains of Vibrio. Okay? The high pH will suppress normal intestinal flora but allow the growth of Vibrio cholera. Vibrio cholera on TCSB looks yellow, and that is due to the sucrose fermentation. So here you can see Vibrio cholera on TCBS agar. Note the yellow colonies of this species on TCBS media. The yellow colonies on TCBS media indicate sucrose fermentation. Now microscopically, Vibrio cholera is a slightly curved gram-negative rod. Now there are a couple of different ways that we can identify Vibrio cholera in the microbiology lab. We can do biochemical and enzymatic testing. For example, we can set up an API. An API 20E will actually code Vibrio cholera. We can also do what's known as the Vibrio string test, where we emulsify a colony in 0.5% deoxycholate and then touch it with the loop. And then what we can do is move our organism further and further away from the slide, and what you can see is a mucus-like string that appears. This is a positive Vibrio string test. We also want to serologically type Vibrio cholera if we do identify and isolate it. To identify Vibrio cholera, one of the first things we want to do is gram stain it. It does have a comma or curve-like appearance. It is a gram-negative rod. It used to be known as comma bacillus because of the comma or curve-like appearance of this gram-negative rod. Metabolically and enzymatically, you can see that it's catalase positive and oxidase positive. Spot indole positive. The TSI is going to be acid over acid because it is a sucrose and a glucose fermenter. The LIA is going to be purple over purple. Indole is negative. Methyl red is positive. Your VP is either going to be negative or weak positive. Your citrate is either going to be negative or weak positive. Urea is going to be negative, and then finally gelatinase is positive, which is interesting because there's not a lot of organisms that are gelatinase positive, but this is one, and Serratia is another one. Vibrio parahemolyticus is an invasive organism, and it is a halophilic organism. This means that this organism requires salt to grow. This isn't super common. There's about 4,500 cases in the United States that occur every year, and we can see that this particular organism causes gastroenteritis, and it is associated with contaminated seafood. So clams, oysters, and crabs and eating things like that can cause a person to get an infection with Vibrio parahemolyticus. This is a water bacteria prominent in coastal waters. Okay, it actually prefers seawater, and seawater has a high amount of sodium chloride, which Vibrio parahemolyticus can tolerate. Okay, this causes a mild to moderate self-limiting illness, and the patients experience watery diarrhea and abdominal cramping. They also experience things like fever, chills, nausea, and vomiting for about 3 days. Okay. The more serious type of Vibrio parahemolyticus infection is associated with the Kanagawa hemolysis. This hemolysin is seen in strains responsible for bloody stools. It's very virulent. Less than 10% of the strains are positive for the Kawagawa toxin. Now in TCBS, Vibrio parahemolyticus is green, and that's because it is not a sucrose fermenter. Remember, Vibrio cholera is yellow because it is a sucrose fermenter, while Vibrio hemolyticus is green on TCBS media. To identify Vibrio parahemolyticus, one of the first things we want to do is a gram stain. You will find that it is a curved gram-negative rod. Enzymatically, it is catalase positive and oxidase positive. On the TSI, it is alkaline over acid because, again, it only ferments lactose and sucrose and not sucrose. It is lysine decarboxylase positive, and it does tolerate 8% sodium chloride, but it will not tolerate 10% sodium chloride. Okay. There's actually a new strain of Vibrio called Vibrio vulnificus. It also causes gastrointestinal issues and wound infections associated with seawater. Other toxin-producing bacteria that are on the list would be E. coli, especially the enterotoxigenic strains, Staphylococcus aureus, Bacillus cereus, and Clostridium perfringens. Clostridioides difficile is the new name for the old Clostridium difficile, but that's the new name. Usually we identify Clostridioides difficile by looking for cytotoxins A and B as the primary cause of antibiotic-associated pseudomembranous enterocolitis. Toxin A is associated with fluid accumulation, while toxin B is associated with tissue destruction. Most antibiotics have been implicated in causing pseudomembranous enterocolitis at one time or another. An actual Clostridioides difficile organism can be difficult to grow, but it's not impossible to grow. You can actually grow it on CCFA media, which we'll talk more about when we get to anaerobes. Okay, if you can demonstrate either toxin A or toxin B, it's enough of an indication to make the diagnosis of Clostridioides difficile. Okay, the toxins are actually identified by fecal ELISA tests. Some signs and symptoms of Clostridioides difficile would be watery diarrhea, fever, anorexia, nausea, and abdominal pain. So let's take a look at how we would culture for this organism. Like I said, this organism is quite fastidious and can be difficult to culture. It is an anaerobic gram-positive rod that has subterminal spores, and it is motile as well. Okay. For culture and isolation, we can use an anaerobic PEA. It should grow there because it's gram-positive. And we're also going to set up a selective and differential media known as CCFA, or cycloserine sulfoxetine fructose agar. That is selective for Clostridioides difficile. We can also set up an egg yolk agar. Egg yolk agar will help us determine if our organism produces lecithinase or lipase enzymes. Okay. On an anaerobic blood agar, colonies are about 4 to 8 millimeters in diameter, and after 48 hours of incubation, they may demonstrate what's known as a ground glass appearance. As mentioned before, it is a gram-positive rod and it is spore forming. The lipase and lecithinase tests are negative. So let's go ahead and talk about egg yolk agar and how we would speciate Clostridium or Clostridioides. Egg yolk agar is also known as McClung Tobe agar, and it is used to speciate the Clostridium or Clostridioides. We can see if our bug produces lecithinase or lipase enzymes. If our bug produces lecithinase enzymes, then we are going to look for a precipitate that radiates into the media around the colony, and you can see that in the top image. That is an example of an organism that is lecithinase positive. We also want to evaluate if our organism is lipase positive. Here, and you can see this on the bottom image, we are looking for oily or a mother-of-pearl sheen on top of the colonies. But by demonstrating lecithinase and lipase activity, we can determine the species of Clostridium or Clostridioides that we are dealing with. Campylobacter is the most common cause of gram-negative rod dysentery and diarrhea. Usually it grows in about 48 to 72 hours. There are approximately 850,000 cases per year in the United States according to the CDC, and about 70 deaths per year associated with Campylobacter. This organism is 7 times more common than Salmonella or Shigella, and it can be carried by turkeys, chickens, waterfowl, and house pets. On the media, they are small, mucoid, gray, and non-hemolytic. They may be flattened out or spread out, and they may actually grow better in the 3rd or 4th streak, and that's due to its nutrient usage. Colonies may stretch out along the streak itself, and you can kind of see that happening in the top image over there on the right. Microscopically, they are a gull-winged or S-shaped gram-negative rod, and you can see that on the bottom image on the right. Presumptively, we could say we have isolated a Campylobacter by first doing the gram stain. The gram stain is going to demonstrate a gram-negative gull-wing or S-shaped gram-negative rod. Motility-wise, and we can observe this especially on phase microscopy, it has what's known as a darting motility. They are oxidase positive and catalase positive as well. Now, when we gram stain these organisms, like I said, they are gram-negative gull wings or spirals. You might want to leave the counter stain on a little bit longer. Sometimes if we counter stain with 2 to 3 minutes worth of safranin, you can see the Campylobacter a little bit easier. Campylobacter jejuni is the Campylobacter species most commonly isolated and associated with a diarrhea type of illness. You will see there that Campylobacter jejuni is sensitive to the nalidixic acid disc, and it is resistant to the cephalothin disc. Now, a less commonly isolated Campylobacter is Campylobacter hyointestinalis. The old name is Campylobacter intestinalis, but you will see they are just the opposite with their nalidixic acid and cephalothin results. Campylobacter hyointestinalis is actually resistant to nalidixic acid and sensitive to cephalothin. Now keep in mind, Campylobacter does not grow at 25, so we're not going to confuse that with Yersinia intercolitica, but it does actually grow best at 42 degrees, and it grows best in a microaerophilic environment. Campylobacter, like strep B, is hippurate positive and can be further serotyped like any other stool pathogen. Aeromonas hydrophilia can cause gastrointestinal symptoms along with wound infections. It can cause any type of infection associated with fresh water. Some wounds treated with medical leeches have been infected with Aeromonas that were colonizing the leeches, so it's kind of interesting. Aeromonas hydrophilia is a gram-negative rod. It's going to be beta-hemolytic on blood agar, so all the time that we're dealing with stool cultures, we're always looking for beta-hemolytic oxidase-positive organisms. That could be a Vibrio or it could be an Aeromonas. Okay? If you have an oxidase-positive beta-hemolytic organism on blood agar, this strongly suggests that you are dealing with Aeromonas hydrophilia. Aeromonas hydrophilia will produce a toxin known as the asaotoxin. Okay, this is an alpha hemolytic toxin that destroys red cells, and this was named after the person who actually discovered the toxin. Aeromonas hydrophilia is oxidase positive and catalase positive. It will reduce nitrate to nitrite, and it's negative for lysine, arginine, and ornithine decarboxylase. Here we can compare Vibrio cholera to Aeromonas hydrophilia, and you can see that the TSIs are both the same at acid over acid. The catalase is the same. Vibrio cholera is esculin and VP negative, while Aeromonas is esculin and VP positive. Vibrio cholera is ODC or ornithine decarboxylase positive, and Aeromonas is ornithine decarboxylase negative. Vibrio cholera is negative or will not tolerate 7 to 10% sodium chloride. However, Vibrio vulvaris will actually tolerate 0.5 to 1% salt. Okay, Aeromonas hydrophilia, on the other hand, is also 7 to 10% sodium chloride negative. Won't tolerate 7 to 10% sodium chloride. It is interesting, Aeromonas will also grow on CIN like Yersinia intercolitica, but it is more likely that anything growing on CIN is going to be Yersinia intercolitica, especially if it's growing at 25 degrees. Enteric pathogens such as Salmonella and Shigella can be serotyped, and we want to serotype them for epidemiological reasons. Okay, now just understand that Salmonella and Shigella have different types of antigens, including somatic antigens, which are the O antigens. These are cell body antigens, and they're heat stable. And then we have the H antigens, which are also known as the flagellar antigens, and these are heat liable, or heat actually destroys the H antigens. We also have K antigens, which are an example of an envelope antigen that surrounds the O antigens, and it may prevent somatic serotyping. Okay, VI antigen is a type of K antigen that may actually interfere with typing of Salmonella. There are a number of serological groups for Salmonella, including groups A through Z. We can do a couple of different serotyping procedures in order to identify those antigens. There's a slide agglutination test, and you can actually number or identify those somatic antigens as numbers 1 through 64. We can use the H antigen as a way to further classify Salmonellas. The H antigen, of course, is the flagellar antigen. Generally, we do a tube agglutination test in order to identify the H antigen. But there's actually two different H antigens that present and occur at different phases within the clinical disease process. So it's important to test our patients a couple different times at a couple different days or a couple different intervals. Flagellar antigens are both numbered and lettered, and a VI antigen may also be present that can be removed before the somatic antigen confirmation can occur. We always want to remove any somatic antigens by heating or boiling and then reserotype. Salmonella has probably one of the most difficult nomenclatures to understand, or a difficult taxonomy. Salmonella, there's two species, Salmonella enterica and Salmonella bongeri. Salmonella enterica has 6 subspecies and Salmonella bongeri has only 1 subspecies. These subspecies are divided into 50 serogroups based on their somatic antigens, and they're further divided into 2,300 other serotypes based on their flagellar antigens. The CDC recommends that organisms be identified as Salmonella and serotype omitting the species name. So for example, Salmonella serotype Typhimurium group B is how we would actually identify or report our organism name. Okay, so the taxonomy for Salmonella is quite complicated, but it is mostly needed for epidemiology purposes, but not necessarily clinical microbiology. All right, so the Salmonella typing procedure here, you can see that we have the antigen. That antigen is on the bacteria, and the antibody is a commercially made anti-sera. And when you put those together, you can see agglutination or clumping. The somatic agglutination appears granular in nature, while the flagellar agglutination appears floccular in nature. And that's really splitting hairs. Granular looks more irregular and kind of chunky, like grains of sand, while floccular looks more feathery and more uniform than granulation. Anytime you go to serotype a Salmonella or Shigella or anything like Campylobacter or even E. coli 0157H7, you could encounter some problems. One such problem is cross reactions with other organisms in close proximity to the organism being tested. This is why we want to make sure we use a pure culture. Our VI antigens may inhibit significant antigen agglutination, and they may be difficult to remove, but you can try to boil them away. Also, if you have chunks of agar from your plate, this could cause false positive interpretations of the agglutination methods. The procedure for typing a Salmonella and Shigella is exactly the same. Okay, we're going to make a very heavy suspension of pure isolates in saline from a new or freshly grown blood agar plate. Okay, we want to make a heavy suspension in a 2 ml saline. Then what we're going to do is take a drop of that and put it onto a slide or a sterile petri dish, and we're going to add a drop of polyvalent anti-sera to a section of that clean petri dish. Then we're going to use saline as our negative control. We're going to mix our suspension with the sterile stick and spread it out, and then rotate the plate by hand for about a minute, and we look for evidence of agglutination. If our organism agglutinates with the polyvalent antisera, then of course we're going to repeat the test using very specific antisera. The polyvalent antisera includes groups A, B, C1, C2, D1, and D2. And basically, if your polyvalent antisera clumps, then it means that it is one of those specific antigen groups. Okay, we're going to report out our result according to which specific antisera agglutinates, and positive results with the VI antigen have to be resolved. And that's what I'm saying. Sometimes the VI antigen interferes with serotyping of your Salmonellas and Shigellas, and so you have to boil those away, let it cool, and retype. Okay, let's talk a little bit about Shigella. The Shigella serotyping procedure is pretty much exactly like the Salmonella serotyping procedure, where we're going to make a very heavy suspension of our organism, fresh organism from a blood agar plate, and we're going to make a very heavy suspension in a 2 ml saline. Then what we're going to do is we're going to take a drop of that suspension and add it to the antisera of the Shigella. And if it agglutinates, it means that it is positive for that antigen. Now, I've mentioned this before. Sometimes we have envelope antigens that make it difficult to type the somatic antigens, and sometimes that can happen with Salmonella and Shigella. And so in order to get rid of those envelope antigens, what we do is we take our organism suspension and we heat it in a heat block, or we boil it in order to get rid of that envelope antigen. Once we get rid of that envelope antigen, we can allow our sample to cool, okay, and then retype it and observe for agglutination and report out the serotype accordingly. Serotyping for E. coli O157H7 is similar to serotyping for Salmonella or Shigella. Okay, many strains of E. coli can produce Shigella-like toxins known as shigatoxins, and those shigatoxins can lead to certain diseases or complications, one of which being hemolytic uremic syndrome. The most common strain isolated of E. coli, the most common strain that causes problems is O157H7. And what that means is O is the somatic antigen 157 and H is the flagellar antigen 7. So that's how it's been serotyped. We can actually serotype to confirm that we are dealing with an O157H7 antigen. We're going to serotype with the O157 latex and then serotype with the H7 latex. If they both agglutinate, then that means that our organism is positive for O157 and H7. To serotype an E. coli for O157H7, essentially what we want to do is have 3 columns of 3 circles each. We want to put our positive control in our first 3 wells of the first row and our negative control in the 3 wells of our second row. And then we put our organism that we are testing in the 3rd row. Okay, you can see that our positive control with the O157 latex is positive, and our positive control with the H7 latex is positive, and it should be negative with our control latex. Our negative controls are going to be negative for O157 and H7 and the control, and then assuming our patient is positive for O157 H7, it'll be positive for the O157 latex and also the H latex. So that's it. That concludes Unit 18, where we discussed our miscellaneous gastrointestinal pathogens and methods for identifying certain things like Yersinia intercolitica, Vibrio cholera, Vibrio parahemolyticus, and other organisms such as Clostridioides difficile, Campylobacter, Aeromonas, and that's about it, and a little bit about serotyping Salmonellas and Shigellas. So stay tuned for Unit 19.